While we're figuring out the logistics, welcome to the folks that just connected. We usually do the class in person, and we will be doing many of the classes hybrid, but today we're specifically just testing a online only option just to see what works. And I'm already seeing some of the folks from the global cohort are here as well. So just give us 2 more minutes and then we'll get the introductions and get started with the. And then Anton, could you also at some point of time record the session for folks who couldn't attend?
Yeah.
Yes, it is being recorded currently. A quick question for all of you that are just joining. We want to make sure that we're set up properly to avoid any Zoom issues. Could, for example, let's see,
Deborah.
Valeria, is there a chance you could check if you try sharing screen, will it allow you to or not?
Like now?
Yeah.
Yeah, it allows me to.
Oh, it does. Interesting. Okay, so
Yes,
we need to disable that.
Too as well.
Yeah, we should disable that.
I'll see if I can do that right now, and then I'll go check for the folks in the room.
Okay, yeah, I think what we'll do is we will get started while other folks are still connecting. Maybe just a show of hands, just so people know the logistics on Zoom. How many of you are in the Stanford cohort and the folks that are joining in the global cohort? So maybe the folks that are in the global cohort, if you could just raise your hands. There is a Zoom way of doing the hands is also if you go in the reactions, you can click and there is a raise hand and you can, and then if folks are okay, because we'll briefly do quick intros, if your bandwidth allows, if people can turn their cameras so people can see each other. I'm already seeing 4 or 5. We today morning sent out an invite to 90 or so students that will be in the global cohort. Clearly that email was sent a few hours ago, so I'm glad some of you saw it. So just I'll back off a little bit. Welcome to Frugal Science, BioE 271. Most of the Stanford students have already heard an intro to what we are trying to do in this class. And I think for the folks that have just been invited, you should take a look at the website and the lecture. Listen into the introductory lecture, which is the first lecture from last year. It's the same or roughly the same framework, just so you have a sense of what we'll be doing in the class. We will begin with quick intros from people from the global cohort. If you could just mention who you are in one line, what you currently do and where you're connecting from. And one of the threads that we'll follow up on is I will do a separate session with the global cohort to introduce you all to the class and the logistics of the class, because I think we are a week into the class per se. And so I'll do kind of a little bit of a catch up for everybody. So while we're still waiting for folks to connect, maybe Erican, Deborah, and Leela, and then Guy, just one line intros.
Should I start, Manu?
Yeah, go ahead, Deborah.
Hey, I'm Deborah Singer. I know Manu from, he's part of an NSF Science and Technology Center, the UCSF, and I'm just really fascinated with what he's doing with this class, and I asked if I could sit in, so thank you. Yeah.
Okay, cool. I think I can go next, right?
Yes.
Erkan Baloku, I'm connecting from in South Africa, so I'm a master's student in molecular and cell biology at the University of Cape Town, and I'm from Uganda, so I'm just here for school. And I was, because when I checked about what Frugal Science is doing, I was really impressed, and I thought I could be part of the class. So, and that's why I'm here. And I'm also part of IGM, so I'm an IGM ambassador to Africa. And I've been doing this work for now 2 years, so I thought I would be here to adapt to the science. Yeah.
Welcome, Erican. Next, Leela and then Guy.
Hi, my name is Leela Mohan Vinati. I work as a Java virtual machine engineer. Completely different background. I did my master's in computer science. Mostly into compilers and runtimes. So this is when I know Manu. I've been listening his TED talks. Great work. Reading his all this update. Just like the other reason also is I have a 7-year-old son and he's doing his second grade now. Maybe this is a good time. Basically bring back my science curiosity and with him. Okay, that's how I'm planning to do. Okay, and thanks for this opportunity.
Yeah, no, welcome everybody that's connecting from the global cohort. We're doing very quick intros, but let's keep those to one-liners because I want to get to the content of the class. And I'll do another session, which will primarily be a meet and greet. But while we are just waiting, I just want to cover some introductions. So Guy and then Emma.
So hey, I'm Guy Edelberg. I'm connecting from Tel Aviv, from Israel, and I just finished a PhD in Paris on democratizing lake acid and isothermal stuff.
Emma?
I'm Emma. I'm joining from Ireland. I'm currently a youth worker with a conservation charity, and I'm currently trying to secure funding to do soil analysis workshops with my students.
Mm-hmm. Ananda? Anybody who's from the global cohort, if you could just raise your hand so I know who all is connecting.
Hi, my name is Ananda. I'm from Toronto, Canada, and I mostly have a food policy, food activist background and have worked in kitchens.
Welcome. Anybody else from the global cohort that's connecting today? Oh, I see Arusha. Yeah, I think that's good. We are all good. Yeah. Anybody else who's connecting from the global cohort? We're just doing one-line intros. And I think we'll continue doing these so people get to know each other. I don't want to eat too much into the time of the class. Okay, you know what? Let's get started. And what we'll end up doing is at the very end, so just as a backing up a little bit on the logistics side, we will run the lectures in a hybrid format. Folks who are not physically on Stanford will always have a Zoom link access to it. We realize that the time zones are very complex, and so many of you might be watching them asynchronously. I'm really delighted that some of you can connect in person, but when you can, it's very valuable to connect online. If you have the bandwidth and you feel comfortable, it's very valuable for the lecturers and the mentors and people that we invite to give the talks. If you can share your screen, but I feel perfectly comfortable if you're not comfortable sharing the screen. But it's just sometimes for connectivity in terms of being able to understand the human beings behind these little pixels, it really makes a huge difference. So backing up a little bit, my name is Manu. We've been teaching this class for 2 years, but we've been doing work in this context or philosophy of frugal science for almost a decade. We run this class primarily with the goal of teaching the philosophy of frugal science, but building cohort and a broader community. So there is a community from last year, roughly around 100 to 200 active students spread across the world. We ran roughly around 30 different projects. There are a couple of just rules of engagement, which are covered in my introductory class that some of you can take a look at. But it's extremely important for just all of us to have a sense of respect for culture, respect for the places and locations many people are connecting from, respect for ideas. We are trying to tackle societal challenges, but with a very open mind. And I think it's very valuable that you'll see that sense of respect and love represented in the Discord channel itself. Much of the communication will happen through the Discord channel. And the folks that are connecting on the global side, I'll set up another introductory Zoom session just to give you guys a background flavor for how we run this class, how we build project teams. Every week we have 3 lectures. One is a technical lecture that dives into a technical field all the way from optics to fluid mechanics to we did back of the numbers calculations this week, applied math. And one of the other threads would be, and the second lecture every week is really focused on a very broad set of challenges. So you get exposed to new types of challenges that we are broadly as a community are interested in. And then there is a lab session, which is an open-ended session. It starts at 1 p.m. Fridays.
Hey,
A lot of
Manu.
demos, past projects we want to share. Zoomcast is. I'm trying to figure out, did I get frozen for a
You
whole
got
bunch?
frozen.
Am I back? Okay. This is a little bit unfortunate. Okay, but I think we have no choice. Let's continue and get started. And I think today's, we have done 3 lectures so far, and they are all online, so you can watch them when you would like. There's an introductory lecture. There is a talk on frugal optics and how do you really build optical solutions frugally. And then there is a talk on what I call by the numbers, or essentially what are called Fermi's problems, which is the idea of estimating numbers essentially. So I'm curious, just before we start, for the homework, anybody in the Stanford cohort, if you would like to just mention if people have actually picked up some of their problems for this assignment for this week. And in that process, some of the global cohort will also realize what the actual assignment is. Anybody from the Stanford cohort, would you like to just share what is a problem for the homework assignment 1 you would like to solve, or 1 or 2? You can just briefly share if there is a problem that you're thinking about that you'd like to. So I'll just describe the homework assignment. We would like you to choose a problem that you're interested in and really dive in quantitatively and estimate numbers that allow you to understand the scale of that problem. So a problem that we discussed last time in the class was news reports of droughts in Ethiopia, for example. We would like you to start by the kind of data that you can collect from our world in data, many of the WHO backgrounds, many sets of data sites that are accessible. But the calculation you're trying to do is also estimate roughly the types of things you might not just be able to find. So we went through the entire lecture trying to figure out how many parameters do you really need to know to be able to estimate the context and the severity of a drought. You know, the number of people involved, what are the water sources associated. And the purpose of this exercise is for people to feel a sense of scale, even at the very beginning of the problem you will try to tackle. So I'm curious if anybody else wants to chime in and mention a problem they're excited about, or certain sets of, you just have to mention the problem just so it's clear to the global cohort how they will be picking up a problem, for example. I'll wait a minute or 2 if anybody wants to. You can just raise your hand. Yes, go ahead. And you can also introduce yourself.
Hi, I'm Susan Zola Pasner here in the lab at Stanford. I'm a fellow in the program at Stanford, coming back to learn new things. I'm an immunologist, and the problem that I posted is how a simple to use diagnostic kit for tuberculosis would decrease the number of TB infections per year.
I have to say, what a fantastic problem. So this is a great example of thinking about asking, when you ask important questions, you get important answers. And what's so beautiful about this is in this perspective is you will be able to evaluate whether this approach of developing a very low cost TB diagnostics that could be rapid is a valuable investment, even if it takes a lot of effort and time and money, because it would have a kind of an impact. And even if you do not know as yet what the kinds of parameters would be, you could estimate it. And the perspective of frugal science would be is in your calculation, you can say if the test costs $1 and a test costs $10, how does that impact the demographics? And so I think, Susan, one suggestion that I would make is try to do that for a specific country. So sometimes context is important, and instead of doing that for the world, try doing this for South Africa or India or US, for example, and you might find completely different answers. And this is also valuable in when you are choosing the problem for by the numbers. I want you to be able to explore this perspective on with a very specific context. So I think with that, I want to get started. Today's talk is really going to be about, so this is the homework. People should do this homework and then share it on Discord. And relationship to Discord, here is the Discord channel. You will all be able to see the welcome messages that have the lots of details. This is the introduce yourself, so folks should actually introduce themselves. And then every time we teach, for every lecture, we create a new channel. So this was the Fermi's problem, and you can see already people are posting. And one resource that I would like to highlight that came about that I had not seen while I was giving the lecture was the Ron Milo, Rob Phillips team have now released a second website that's called Anthropo Numbers, which is essentially very much like biology by the numbers. Some of you remember this website that I had shown you before. Biology by Numbers started as a website to very quantitatively think about cell biology with, you could ask any question. Now they have human impact by the numbers. And what's beautiful is I could ask how many cows on the planet, and oh, no match. Let's be more scientific. Livestock population, and standing population of pigs is 1 billion. And it's valuable for you to kind of play with these types of resources because it'll give you a context of the problem you're trying to work with. And what's nice is that it will tell you what the source is, and then you can follow the source and go along the way. So this is a fantastic resource. It's very timely. The bioRxiv paper just came out. Okay, so I'm gonna switch. And I think, Tyler, if you can manage letting people in now, because my screen kind of gets lost. So today's topic, we're going to cover the oceans, why we should care, and what types of problems are actually very important and are completely missing. And then I'll end with a case study of an example of a project that we have been working on in this space. But I think every week we will cover a problem domain. And so the problem domain today is the ocean itself. Some of you live next to the ocean, so you have a sense of both mystery and pride and a sense of vastness of the ocean. And some of you might never have seen the ocean, like I was when I went to college the first time. And it never occurred to me how big the water world is. So to put that in context, when you ask yourself, how big is this ocean and why should we care? We are terrestrial beings. One of the ways of thinking about this is 70% of the planet's surface is water. It is one of the largest biosphere on the planet. Half of the oxygen that we breathe comes from the ocean. You can think about the heat that's absorbed. 93% of all of that heat gets absorbed by the ocean. 50% of CO2 that we emit as human beings is essentially absorbed by the ocean. It is full of life. It's incredible in terms of the living context and both the microbial and the larger biomass that's present. And one of the perspectives of thinking about it is, ironically, with all of this, we know very little about it. And I think the common framework that people often use and say that we sometimes know more about the surface of the moon than the bottom of the ocean. And you might think, oh, that cannot be true. How is that actually possible? And it turns out observing the ocean is actually quite difficult. And we're going to spend the first 20, 30 minutes of this talk trying to understand why that is the case and why it's so easy and simple, relatively speaking, to point a telescope to the moon and make surface measurements of the moon. You know, the moon it's a dynamic system that it's rotating around the Earth, but there is very little dynamics that's going on the surface as far as we know. And what I'll try to convince you is the ocean is a living system. And as is true for all living systems, the complexity scales astronomically. And that's why it becomes very difficult. And we are, you know, it's 2022 and it's still hard to say because a very small percentage of the ocean has actually been explored. Much of what I'll talk today is sets of conversations and a slide deck that Emmanuel Boss, one of the mentors of the class who will join at a later time, had put together last year. Emmanuel and I have been working on a few new projects in the context of the ocean. And I think I've modified this lecture also very focused on the kinds of problems that we are trying to tackle as a lab as well. But I think, yeah, really lots of credits goes to Emmanuel for teaching me a lot of challenges that exist in the ocean itself. So one of the contexts of this is we want to think about oceans not just as a biological curiosity, but we want to also think about this in the context of its interface with the humans. And often enough, many of you have heard this term planetary health in the scenario where folks might be thinking about that the health of the planet is as important, and we have to think about health in a systems perspective. And the sets of questions that we will really focus quite a lot today on is how do we currently measure the ocean? How should we measure the ocean? And most importantly, what does frugal oceanography look like? Why you could essentially change what we have been doing so far and where we have gone wrong in terms of these types of measurements. And I'll very specifically talk about one project, Planktoscope, that we've been scaling as a lab, which is a frugal oceanography project. So, you know, I already mentioned much of this for the context of thinking about the ocean. One very important aspect of this, which has been labeled as the blue economy, and it's a little bit tricky to think about. I think from a food resource perspective, it's an incredibly important scenario, roughly around, 282 billion, for example, is the US ocean economy. And one of the perspectives there is 3 million people employed. You can also think about the vastness of the incredible population that literally lives on right next to the ocean. And so their livelihood also depends on it. And the other aspect, which is a little bit troubling, which we can briefly talk about on the blue economy side of the story, is this notion of being able to exploit and extract resources that exist in the ocean. One of that is being talked about currently is deep sea mining. Because we do not know the ecosystem, the benthic ecosystems that grow on the bottom of the ocean, it is quite troubling to think about if we start exploring and exploiting an ecosystem that we as yet do not understand. So we'll talk a little bit about that as a very specific problem. But let's just do some kind of a back of the envelope of the common things that you hear in the news. First one is oil spills. This is a huge impact when you think about what we have done in dramatic context as humans to the ocean. One perspective, if people want to read this 2011 paper, is a summary associated with how oil spills are actually still underreported. So you might think that you might see all the oil spills that happen, but many times some of these leaks happen in oil rigs down below, and it's quite late until a point that you actually realize. The other thing that has been talked about quite a lot in 2021, which is this notion of, you might argue that these are accidents, our global economy depends on it. How could we actually predict? Ironically, there are threat assessments on specific locations. And one of the biggest one that people have talked about is the SFO Safer. This is a rusting oil tanker off the coast of Yemen that has been sitting for many years. And most scientists believe that this is a catastrophe waiting to happen, primarily because much of it has kind of been abandoned in some sense. There is 3.1 billion barrels of oil that this has the capacity. And while this is just sitting off the coast, it's an offshore filling station. Because the infrastructure has deteriorated so dramatically because of also the political situation in Yemen, many people believe this is an environmental catastrophe waiting to happen. And still, after all of this coverage, there has been very little action. I think the thing that I want to highlight with this is this notion that sometimes we believe that our actions are unpredictable, and these unpredictable scenarios lead to these catastrophes. But you would argue, and I think something that has been said for the fires that happened in California, is that all the signs were always there. And so you can, and you have the capacity of applying scientific approaches to bring the kind of oversight that's actually needed on these types of infrastructures. The other context of this is historic. If some of you are interested, as you're spread around the world, find the largest oil spill closest to you. I remember just as a child, the oil spill that happened in the Gulf War and certain sets of oil spills from the past deeply impacted me just in the pictures that I saw. And I remember as a high school student working on projects around oil spills. So I think this is very dear to my heart. Of course, the kinds of solutions we were thinking about then and now, I find it ironic that not much has changed as dramatically as we would like to see. The other factor associated with human context is eutrophication. This is something that some of you might have seen if you live in coastal cities, which is the runoff of inorganic nutrients. And one of the things you might argue and think about is, if you have a runoff of nutrients like fertilizers and nitrogen out into the ocean, it must be a boom, because it creates a massive amount of bloom of plankton and life in the ocean. The challenge is that suddenly that peak, when it dies, it settles to the bottom, it takes away all the oxygen, and it creates what are fundamentally called in the ocean dead zones. So what I'm showing here are just these red spots. These are all the dead zones in the ocean. As you can see, they occur everywhere, and they also occur where there is highest human activity on the coastline. Now, the capacity of being able to measure and quantify the impact of dead zones is very limited, primarily because some of this is happening on the coastal context, but it's also happening farther away from the coast. We can measure the kind of toxicity that we actually see in higher animals in the ocean, but really from a microscopic scale, it's quite difficult to measure. And I'll talk a little bit about challenges around measuring dead zones. You can also think about these dead zones also in aquatic water systems. I'll cover a lecture on water a couple weeks from now. So much of this is with the lens to the marine context, but there are deep connections between, if some of you are interested in problems in the water system, especially freshwater. Of course, this one, I don't even have to talk about. This is plastics. All of you have heard about it. It would be fantastic to explore certain class of projects in this space. This, in some sense, galvanizes a lot of people But I think one of the things connecting with the missing fundamental science, one of the irony of this problem is, as I was saying in one of my early lectures, is only 1% of the plastic in the ocean is accounted for. So 99% of that is missing. And so, of course, all of you have heard about these garbage patches, the Eastern garbage patch and the Western garbage patch. One thing that we realize is this is only 1% of the plastic that's in the ocean is on the surface. And I'll explain why that is the case. But 99% of it is completely missing. And that sets this challenge of how could you possibly come up with a solution associated with this if you don't even know where to identify it. This has been a big scientific mystery. Now we know and understand that much of this gets assimilated in microplastics. And this type of a plastic has actually made it to the deep ocean. So the picture that you're looking at the very left is actually from deeper waters. And you start realizing that much of this, because plastic has a certain density, either it floats or it sinks, and eventually it assimilates itself in the volumetric context of the ocean, which is quite bad news because then we really don't have many ways of removing that at this point, especially when it comes to microplastic. So the biggest factor is if we could stop entry of plastic into the ocean, that really is where we should concentrate and think significantly. The other aspect of this is when you think about what we put in the ocean, and plastics is a great example for this. The ocean has a density fluctuation, which is vertically stratified. So roughly it's a 5% change. Seawater gets denser and denser as a... So you could see what you put in the ocean will find a very specific height and then become buoyant at that height. So many of these solid pollutants, if they are heavier than surface water, will go deep into the ocean, and then essentially you really have no access to them. Although they're still impacting the ecological context of what's the life that essentially lives in that in the ocean. Another one, which is a very common one, very well understood, but very little has been done on this front, as a problem if certain sets of folks are interested in from a sustainable materials perspective, is ghost fishing and fishing lines and threads that in terms of the plastic pollution that goes in the ocean, this is a remarkably high number compared to when you look at your plastic straws that you use at Starbucks. And because some of the fishing industry is subsidized, there is less pressure on converting into practices that we would like to transition that are almost necessary as compared to certain sets of things that might be right in front of your face, but might not have a bigger impact. So if anybody is still looking for a problem for the by the numbers, try to measure by weight the amount of plastic that we deposit in the ocean in abandoned fishing lines and compare that to the amount of plastic that we manufacture for the sipping plastic straws. You know, just as a perspective, it will give us a sense of thinking about how large these problems are. The other thing that I just want to mention is related to heat waves. We will talk about much of this in the context of the climate lectures when we talk about climate change very specifically, and I'm going to ask a friend of mine to join that lecture as well. But there is a perspective of this because I said 90% of this heat is actually absorbed by the ocean. We are seeing massive anomalies in heat temperatures and peak heat temperatures locally. And some of you who have been following the coral reef work would realize that this is a huge impact in terms of thinking about much of the ocean life that has not adapted that well to be able to handle these types of heat anomalies. If some of you live in these sets of zones, we'll eventually also talk a little bit about thinking about people's individual perspective on the context of the climate. But I'm going to leave that for the climate lecture because the fact that we are globally distributed would actually be quite valuable in terms of thinking about what we want to do from our personal perspectives. So I think you might be seeing that I'm listing through a whole bunch of problems that as humans have created. We're going to transition a little bit to the biology of the system now. And then I very quickly just want to highlight and transition into the types of challenges that I would love for folks to think about as a seed ground. So for folks new to the ocean is alive. And some of you have seen that by looking at the humpback whales and the fish, but many people have not actually seen the microbial life. So we will keep using this term quite often throughout the lecture, benthic or benthos being life that's all the way at the bottom, nectin being life that doesn't care about the ocean currents, so it's able to just swim wherever it wants to go, for example, whales or fish. Plankton is microscopic life forms that is thought to be at the mercy of the ocean. As a lab, we've been discovering over the years that actually that terminology is wrong. Even the smallest single cells are choosing where they want to be in the ocean, and Newson being the layer that's really on the surface. This is a very important layer that we don't think about, because microbes in the Newton layer might really be seeding the atmosphere for what leads to rainfalls, because the amount of droplets that are generated from shear on the surface of the ocean would lead to atmospheric context. So the atmosphere talks a lot significantly to the ocean, and we'll talk about that in the context of the climate. And at that same time, the other way around, the ocean talks to the atmosphere. And so for some of you who have seen or followed through, for example, the dust from Sahara making across the ocean, or very recently, for example, microplastic from the ocean making it into the atmosphere, and then eventually atmospheric microplastic being also present as one of the pollutants that we did not think about before. And of course, all of this is important because everything eats everything, and much of this gets deposited into the food web. We have very little methods to measure this biology, even to an extent of what eats what in the ocean. So these food webs have been made from an observational standpoint. There is a lot of genomic work that still remains to be done to truly establish the food webs in the ocean, which is important if we are going to think about this as a system. So if some of you are interested in microbiomes and the work that's associated with genetic markers to be able to tease out and automated sets of systems to build food webs is a phenomenal space to be thinking about, and especially natural food webs and food webs as they are changing in the ocean. So what's so different about ecology in the ocean and the ecology that all of us live every day? The ocean is three-dimensional. Unlike all of us, we live a very flatland life. There is a context of thinking about the currents and the winds and the amount of flux that exists in the ocean. But the most important aspect is the ocean remains unobserved and unobservable, primarily from space and satellites and other tools that we have used in the past in a terrestrial context. This is a huge challenge in terms of thinking about what we can do just because of the size. And then all the length scales matter, which is also true for terrestrial life forms because the microbial length scale is very important. And I think what I'll emphasize, at least from the context of the tools and some of the demos that we will do in the lab lecture this Friday, will also be associated around tools that we have been developing to measure the microbial context of the ocean. So I think this is something that I'm going to now skim over. One of the obvious thing, unless you can convince people that the ocean is so deeply connected to our planet that we should care about it, there is also a human health risk associated with it, which is related to toxic algae. If some of you are interested, this is another big problem that still remains in terms of mapping and measuring toxic algae in our environments. There are certain classes of algae and microbial organisms that produce really terrible toxins. And these sets of toxins have tremendous cost on human life, but also on the exports. And it really undermines the context of thinking about the blue economy as well. And I'll tell you a citizen science type of an approach. We got introduced to this problem of toxic algae by an invitation from a friend of mine, Jose, and two of the students in the lab went to Chile. And we learned about this problem, which has been happening for a while, but that led to riots across the country of a massive series of toxic algae blooms across the coast of Chile. So if some of you know the topography of Chile, it's almost a stick with an incredible amount of coastline. And one of the challenges associated with that is once you have that coastline, life depends on it. And salmon farming began in the ocean. That led to a massive amount of use of antibiotics and many other factors, very much like how we've been growing cattle farms and other things in the context of livestock. And the idea that Jose came up with at that time was, could we use, he works for the Ocean Measurement Society in Chile. This is a government agency that measures the toxicity of the ocean water across all of the fjords. And it's almost impossible because you cannot have enough number of people to sample such a vast coastline. And the program that he's been developing is using citizen scientists to track plankton and toxic algae. So this has been growing as a community. There's roughly around 35 schools. And again, going back to some of the context that I was talking about in my opening lecture around citizen science, it's a really fun way of thinking about bringing science to the students. And this is literally data collected from some of those sets of students in terms of thinking about, with very, very simple tools, could you actually detect pathogens that are toxic to humans? I'm going to switch to the notion of what are the current technologies. And you might, I'm going to probably stop very soon after I cover some of the technologies and before I talk about some of the projects from the lab to turn this a little bit into a discussion. But if you have questions, you should also just type them in the chat. And then Tyler, if there are certain sets of questions that come up, either you or anybody else can interrupt me, because the purpose of this is to not be a monologue, but I'm rushing a little bit because I want to get to a discussion around possible projects. So I want to cover some of the basics, but please feel free to type in and write in the chat some of the questions you might have. So let's get back to what is the most sophisticated measurement system that has been developed for the ocean. As I mentioned, 97% of the planet's water is in the ocean, 70% of the Earth's surface is in the ocean. How do we really measure it? Satellites just don't cut it. And the thread around this is really been, so far, we have been doing satellite-based tracking, and you can do a significant amount of work. You can be thinking about, so chlorophyll is measured directly through satellites. You can do that at a fairly high temporal resolution. This is associated with the ocean color. And since 1996, we have a very good data set of the ocean color that's associated with these sets of satellites. Now from those sets of environments, you can also just measure temperature, you can measure currents, you can measure waves. There are certain sets of scenarios of really thinking about the smaller scale. But one thing that this misses sometimes is very much the microscopic life at the smallest scale. This is also the measurement only of the surface of the ocean. And in terms of thinking about this notion of depth, so this is sort of the color data as a movie. And I just want you to also notice how dynamic this system is. And we will talk quite a lot about why measurement technologies in the ocean need to be frugal so they can be distributed, because you have time dynamics. You have to really worry about how these times are changing. And then it's not a static system to begin with. So you need to sample it significantly. This is a technology that I'm quite excited about, and especially in the context of thinking about what does a frugal version of this looks like. I don't know how many of you have heard, this is called Argo floats. It was a brilliant idea that came about in terms of thinking about floating gliders. And I'll talk about why I am enthusiastic about this as a tool. And you can see roughly around 5,000 of these Argo floats are literally floating in the ocean at this very moment. Now, what they have as a capacity is quite remarkable. It is a platform, so you can put many kinds of sensors on it, But rather than just being on the surface, they have what is called a buoyant engine. Now, that buoyant engine can change its density, and hence it can allow you to go deep into the ocean and then come back. So you could drift, from a Lagrangian context, you could be with a packet of water, you could remain on the surface, you can go down for 5-10 hours, get down to 1,000 meters, make recordings of temperature, chlorophyll, whatever sensor that you've put on it, go all the way to the bottom of the ocean, and it has enough battery life to be able to last for a certain amount of time. And then it can come to the surface and transmit that data via satellites. Now, this is where costs come into play. One of the challenges, and again, this is, although I absolutely love this technology because this has really changed the game of what we know about the ocean in real time, it is also a little bit about throwing your money in the water. And what I mean by that is, now look at these cost numbers. The core cost of a single Argo float itself is $20,000. That's just a buoyant engine to begin with. Then you start talking about many sets of assays, the types of sensors, larger battery life. And when you start asking the kind of management program that you have to build around to be able to deploy these, any sets of program can fairly easily get down to around $100,000. And this is one of the challenges in terms of thinking about as a challenge for some folks, if you're excited about the ocean, is to rethink the notion of passive ways. Could be use biology, could be use many other mechanisms to really strip the cost of these Argo floats and still be able to have the same fidelity of the data. I think in the end, this is incredibly valuable for the type of data that it provides. But on the other hand, they are limited. And again, I think one of the issues that is a tricky one to think about, I don't have a good viewpoint in it, but I just want to raise it, is they are not recaptured. Once they die, they sink to the bottom of the ocean. So from a cost perspective, unlike rockets, which we are trying to recover now, we can land rockets back. Could you imagine a passive system that when they die, there is a sense of recovery? There are a couple of really fun ideas around this that are floating around that I'll kind of discuss in lab sessions, but just as a thread to think about. If you want to play with this data and actually see this data in action and try to understand the dynamics of this data, you can go to the Global Ocean Observing System website. You can choose an Argo. There is also a type of a citizen science program about adopting a float in which you get to actually interact and very specifically look at the data. And you're also supporting some of the scientific missions around these. But what are we missing? What we are missing in this context of thinking about the physical, what I mentioned so far are mostly physical parameters of the ocean, other than just chlorophyll. What's completely missing is the biology of the ocean. And I absolutely love this project. It's called the Continuous Plankton Recorder. And I'll tell you a little bit of the history of this project. But it is designed around thinking about capturing the mesoscopic, so not the microscopic world, but the zooplankton and some of the plankton in the ocean. And it was a brilliant brainchild of Alistair Hardy, oceanographer, who in 1930 came up with this idea. And I just want to show you a quick video to just think about how brilliant this is. And, to me, it's a pinnacle of thinking about frugal science. Of course, we as society have been doing frugal science all this while. Hardy came up with this idea in the 1930s to think about a continuous object. This is the object that he created, that you can put not on research vessels, but on shipping containers. So all the vessels that are in the ocean that are carrying our stuff, like when my kids want yet another soft toy that's coming from somewhere around the world on a boat, this little thing gets towed around behind the boat. And I'm just going to show you all a quick video because it's kind of nice to see it in action a little bit. I have to watch this ad.
Plankton are considered one of the environmental drivers. So they're kind of the link between what happened in the ocean in terms of water chemistry, temperature, the kind of water conditions, and the fish, because plankton respond to their environment really quickly, and fish feed on plankton and larger organisms feed on fish. So the plankton are the link between the oceanography and the fish. We know that plankton respond really quickly because they have life cycles that are really short, sometimes even days, but all of them less than a year, or at least a year is the longest life cycle. So if changes happen in their environment, they respond quite quickly. You can see that in changes in their numbers and the types of plankton and where they're at. So by monitoring them, it gives you a really rapid response to a change in the environment. In the early part of the 20th century in the UK, it was kind of hard to know where to send the fishing boats, where they were going to find the herring. And Alistair Hardy, he invented this instrument that could be towed behind ships, measuring the plankton, and it's called the continuous plankton recorder. Continuous because it, rather than taking a sample as a snapshot across, it continuously samples the plankton as it goes. So his idea was that if you could understand continuous, because
so I think I'm mostly interested in showing you the geometry of this object. If some people in the class are actually interested in making a version for this, this would actually be really fun. But the trick and the beauty of this idea is it's essentially two silk screens. It's a tiny sheet of screen of silk that is rolled up, which is what you see in green. And the red is essentially a covering tape that comes on top. It's also another type of a mesh. And it just continuously, from the thrust of being pulled, rotates. There are no batteries, there is nothing. It's just a hunk of metal with this roll. But then this roll traps, so if you can see my mouse, it traps the plankton right here and they get squeezed, and you have recorded a fossil. Fossil because you can take this roll and then bring it back. And then even now, almost 70 years later, I could go back. I could go back to a very particular date and a particular location in the history of this project and actually identify what was in the water at that time. And it's a remarkable sort of a thread. And I think one thing that I've been very excited about this in the context of many projects that we do in the lab is this notion of, even though sometimes you might not have a cost-effective technology to measure the resolution that you're interested in, making these man-made fossils allow the future generation a chance to understand what was going on. So the context is, if you can, you might argue that, oh, this is generating a massive amount of rolls and imaging was not automated at this time. We have been looking at building a system like this that also does automated imaging. But one of the contexts there is that in the past, although automated imaging was not there, but it's there now. So as you're thinking about many class projects, sometimes you might find yourself in this space that I don't even know what to do about this problem because the cost of current measurement technologies are way too high. You could really start a project that initiates something for the future because you're banking it for the future. When the sets of technology prices would go down, people would, another way of saying this is we are living in our lifetime the fastest change on our planet. So we should be preparing for understanding this change, because change, if 10 years from now we see something, could we go back and actually explore? And the cost of creating the fossil can be incredibly low if you design it that way. So anyway, if people are interested in that space, this is another space from a project perspective that I'm very excited about. And one example of this, the way that we have been creating fossils, has been just museums. If you think about the museums, there are certain ocean samples that are collected. And this was a great paper that came out. This is literally the HMS Challenger. This is 1872. And you are looking at forminiferas. Forminiferas are these beautiful plankton that biomineralize a shell. And look at the picture on the top and the picture on the bottom. This is from 1872. This is from 2009. And a foraminifera collected from the exact same location in the ocean. But you see how thin the shell is? And the thickness of this shell, essentially, and they did statistics on this to show that because of acidification, many of the planktonic snails and many of the systems that use biomineralization, because of the acidification, the chemistry of biomineralization is changing. And from these and the statistics, you can actually predict what the pH of the ocean was at that time. Another one of these that is my personal favorite because it happened to me as an accident is a diatom collection in Santa Barbara. I found myself in Santa Barbara at UCSB with nothing to do for a couple of hours, and I accidentally walked into a museum, and what I was drawn towards was a diatom museum. And I got intrigued. I started looking at it, and it turned out there was an individual, a marine biologist named Robert Holmes, that then has basically donated this diatom collection. This is roughly around 6,000 samples from across the world, collected since the 1930s. And he started collecting diatoms at the age of 12, and wherever he would go, he would collect diatoms. And literally, the third slide I pulled out, and nobody has actually used this collection, and I had my little microscope, and I had 10,000 samples to look through. And then the third sample I collected, what really ingrained in my memory was it had a location in the ocean, and that diatom was collected from the surface of a sea otter, and it was listed as a diatom collected on the skin of a sea otter. And, you know, one of the contexts of thinking about these types of things and these types of fossils is now we have the kind of automated technologies to go back to these databases to really try to understand what has happened to our oceans from the past. Because we have a good intuition for the physical context, you cannot make the kinds of models for the biological context. So any of you who's closer to Santa Barbara or people who are excited about a trip, I've been thinking about this for a while, to be able to at least digitize this fossil collection that exists. So let's just also very briefly think in terms of why is it so difficult and what has been done so far to measure life in the ocean. So what I'm showing here is an example. If you can see this plot, first of all, it's a log plot. So you can essentially see, let me just remove my controls. So you can see on dimension all the way from viruses to jellyfish. And here is the sets of technologies that currently exist to measure biology of life in the ocean. Some of the common ones that I'll show you are called flow cams because it's related to the project that we launched. And the bars there show what you can measure. And then, of course, the smallest is flow cytometry. So if some of you are cell biologists, you all know we use flow cytometry all the time to really measure the smallest scale to even an extent that you could really do viral densities and other factors. But flow cytometers are incredibly expensive again, all the way to Coulter counters. Most of these technologies were developed in the biomedical context and then used in the ocean. And one of the most beautiful stories that I love about exploiting technologies that were developed for one domain but applied to the other is the famous story of the discovery of the most abundant microorganism on the planet, which was discovered by Penny Chisholm. It's a type of a microalgae. It's less than a micron, but in abundance, it's basically the highest density and is known to create essentially massive amount of oxygen, controls a lot of the oceanic chemistry. But because it is submicron, for hundreds of years, people had been looking and everybody missed this organism. And Penny had this idea, why don't I literally borrow a flow cytometer from a friend of mine? She had a friend, they were a cell biologist, and she literally took that instrument on the ocean. And for the first time, they were essentially able to record the data sets. And then, of course, they have championed the notion of using flow cytometry as a way to record life at the small scale in the ocean. One of the things that's associated with this comes to also in the context of thinking about microscopy and the overlap of microscopy and sequencing. I'll talk a little bit about this. We have tremendous technologies now to scale up the measurement on the sequencing side. but we are still missing in terms of being able to do the microscopy front, which is what planktoscope does. But there is also context in terms of thinking about being able to link these types of data sets. So, okay, in kind of a conclusion, before we open for a little bit of a discussion, I think what I want you to think about is the ocean is exciting not just because it holds the key to most of the atmospheric and sets of climate change parameters that we think about. Very simply speaking, it's very strong hold on the carbon in the atmosphere. It is also a source of mystery and solutions in the sense that most organisms that we find in the ocean cannot be cultured. Most genes that we have discovered, more than 50% of genes that we have discovered in oceanic organisms have no known phenotype, no known function, because their biology is so dramatically different. And it is not just about, because it's structured as an ecosystem that is continuous, it is much more focused around being able to think not just what exists, but what is it doing dynamically. And I think one of the things to be thinking about from the context of dynamics is what I'll mention in a second, which is life is not static in the ocean. So this is a beautiful animation to think about of the largest biomass migration in the ocean in the world happens in the ocean. So it is not the Serengeti, although how spectacular it is. This is life moving up and down in the ocean, literally by hundreds of meters to even kilometers every single day. And when you do the sets of numbers, what I love about this, going back to the sets of calculations, the amount of energy plankton consumes in moving up and down every single day is essentially equivalent to the energy budget of entire United States. And so you can imagine this is, from a biological context, a phenomena that literally exists, and they spend this tremendous amount of energy. And so if some of you go diving or love the ocean, I don't know if any one of you have done what are called dark dives, which are or night diving. If you find yourself, you might end up diving into a place like this. And this is just to give you this visceral visual sense of how dense life in the ocean actually is. And this is also why the carbon flux and the connection of thinking about how the sequestration happens, which will kind of deal with the context of the discussion itself. The other thing that I just want to mention associated with this is the reason we are so interested in being able to make these microscopic measurements is to think about combining with the kinds of modeling and top-down approaches. So if some of you, I remember I had shown that picture or that paper from the IPCC reports where it highlights the missing carbon, where when you write down the sets of models that we know for how much carbon should be in the atmosphere and how much should be taken by the oxygen, if you do that one way and then you do it the other way, and then there is a gap. And this is also why we need to be able to, in our climate change models, introduce biology. And you can only do that if we are able to actually make measurements in the ocean. One of the perspectives of this is every single thing about the ocean changes because of climate change. If you think about temperature, if you think about pH, when I mentioned that the shell of the foraminifera is thin, that implies that the ballast, which is the real reason that these things sink down and take carbon with them, their sinking velocities dramatically change. So if you account for pH changes in the ocean, you would actually reduce the carbon flux that the ocean is taking and absorbing from the atmosphere. So remember that number from the first lecture that I had talked about, that if somebody switched off the ocean, the carbon sequestration from the ocean, the CO2 in the atmosphere will jump from 400 parts per million currently to 600 parts per million. And this is the context, and this is the biological reason that much of this life effectively sinks back. And then another kind of a context to really think about this is, which I'll mention a little briefly in a second, is oceanography has had its challenge because access to the ocean is complex, but much of the technologies that have been developed have not been built in the context of a frugal framework. So every little instrument and thing that I'm talking about still costs an incredibly large cost, and hence very few people have the context of being able to engage in in oceanography, and especially biological oceanography. And ironically, when half of humanity lives around 200 miles from the coast, it is still perplexing that if on an average, if you ask a person walking on the street that how many organisms or microorganisms might exist in a glass of ocean water, that would be a very different answer, just because we are so decoupled from it, primarily because we don't have the sets of measurement technologies. And I think the thing that I want you to take away from that is, of course, there are a very large class of problems. The thing that, at least as a lab and what we are interested in, and it fits the framework of frugal science, is how do you engage half of that humanity in engaging in both making measurements and bringing the kinds of mitigation strategies we need to engage in problems in the ocean. And that is really where citizen science and approaches of bringing these sets of tools to a broader group of people come into play. So what I'm going to do is I was going to transition to technologies that we have been working on and sets of solutions. But before we do that, I am going to take a little break in terms of questions, discussion. I think this was a little bit of a rapid fire overview for all of you. Some of you, actually, let me just ask this question. Anybody here has background in oceanography? Have you had the chance to kind of do much more of a deeper dive? Or I'm sure many of you have interest in the ocean. Any of you dive, or are any kind of true divers in the crowd, or divers at heart? Yeah, I think I'll wait. It would be valuable if you all turn your cameras on for a second. If people have certain sets of questions or much more, puzzles that you've been thinking about, this would be your time. Before, I'm going to very briefly show you all one project that we are trying to scale, but it's valuable to take a moment if you have any questions on what I discussed so far. Yeah, you can just unmute yourself or raise your hand using Zoom.
I have. Oh.
Go ahead, Erica.
Oh, should I? Thank you. I was going to ask a question about the balance between upstream intervention and downstream intervention in terms of oceanography, because I know that there are projects right now that look at like heat resistant corals and like trying to, genetically engineer different organisms to start working with and being able to live in what we've already created, the environment we're already creating, opposed to like cleaning up the environment. So I was just wondering if you have anything to say about downstream versus upstream.
Yeah, no, I mean, I think, remember, I'm covering a massive space. We could spend a year on these sets of questions and puzzles, and there is an incredible amount of mitigation strategies and incredible sets of ideas that people are thinking about. I think this downstream and upstream is much more of a personality thing, what you choose. Sometimes some people argue that, oh, it's already too late. As long as you are thinking about these sets of problems, I think that is what I would argue, that we have no choice but to think about these problems. It is the harsh reality that we will live in a world that will be different from what has been in the past. And very specifically, today I'm only covering the ocean, and I think climate, I'll bring a couple more people to talk about this. But just from the ocean perspective, the ocean has changed because of us. We have to accept that as a fact. And so the search for hardier coral reefs, for example, or coral species is very valuable, because we might go to zero coral otherwise. And I think in the Red Sea, for example, there are certain examples, because the Red Sea is incredibly hot and you might find species. So the upstream versus downstream is, you could do some of the numbers. I think, for example, the sets of projects around cleaning the garbage patch is such a small portion. I think I often think about this in the perspective of the investment And much of carbon sequestration technologies, when we will cover them one at a time, the question is not that there is an interesting technology. The question is, does it scale to the problem you're thinking about? So I would draw a quadrant, upstream, downstream being one axis, and the context of scale being on the other axis, and cost. Because, I think the challenge right now is, although there is an incredible excitement from the investing community in ocean-based solutions, they are not doing the by the numbers assignment. I've read way too many proposals that when you do the math, absolutely don't make any sense. But still, we are grappling for the straws. And by not doing this type of a deep dive in the science of these sets of problems and understanding the biology of the ocean simultaneously, we're trying to perturb and revive back a system that we as yet don't understand. And so I think the way that I would create that would be for your own self. Like microplastics is a great example of upstream versus downstream. Should you be working in cleaning up the ocean, or should you be thinking about the blocking from the rivers? Because the majority of it comes from 10 cities in the world. You could argue that if you go in, and wherever it is for the global good to build filtration plants right on some of these freshwaters across those cities as a global effort, just like we built LIGO, or just like we built the particle colliders, that would do more as compared to what we might do essentially by sending ships out there. But again, it's a matter of calculations. And then I would say do the upstream, downstream, and then the other axis being either cost and scale. I see another question from Leela. Go ahead, Leela.
Okay. Thanks for the presentation. So I'm coming from a completely different domain. My questions might be very naive. So one of the instrument which was regarding the fossil of the ocean was collected the thickness of the shell, if I understood correctly. Mm-hmm. So this essentially saying how much of carbon dioxide consumption changed as the years passed, and in fact the opposite of it, actually the amount of carbon dioxide produced to the air was increased as the years passed. Mm-hmm. So basically it is dying. It's kind of double. Basically, there's more production of carbon dioxide, but the reduction of carbon dioxide consumption by the ocean. So basically, my question is, so as the terrestrial beings, it is really tough to teach humans how to be conscious about usage. And if you can get this math, what is the balance? So as this carbon dioxide is growing up, is increasing, telling people to, okay, you have to be very conscious about your carbon dioxide production. It's possibly easy to tell the ocean, or increase somehow so that ocean can take more carbon dioxide, which is, I think you said 50%. If you can increase that to more so that this can be balanced. I think my understanding is we need to create that balance.
Yeah, so I think I see your question. Just a few sets of comments. I think today I'm just presenting the context of the sets of challenges. We will cover an entire lecture on carbon sequestration technologies and the frugal science world of it. And in that, I will cover a little bit about what people have been currently thinking about using the ocean and increasing its capacity. There are lots of challenges there. It's not so straightforward. Remember, the perspective of what you don't understand comes back and bites you. So currently, the reason the situation is so troubling is we do not understand the biological pump. We do not have a first principles understanding of this sequestration that has really been saving us for many of these years without knowing us, without us realizing. And so the challenge there is an intense amount of efforts in being able to understand. I mean, right now, there are completely competing hypotheses. We are working on a hypothesis with a few sets of collaborators that oceans have pandemics all the time, just like the pandemic that we saw, and that viruses play an incredible role. But the challenge is we cannot even confirm as yet whether viral infections in the ocean in these populations actually increase or decrease carbon fluxes. You could argue for both, and the measurements really are missing, and that's really where we have to go. So we'll talk about sequestration quite a lot and then think a little bit about this. I think the perspective of the thickness of the shell was just this notion that time has passed and we did not have certain sets of technologies to what was happening in the past. So looking in our past is quite valuable because all of these fossils and sediments, essentially when they sink, they deposit. So you can go to the bottom of the ocean, do a sediment core, and from that core determine 10,000 years ago, 20,000 years ago, an organism that was alive, and use that as a recorder for the chemistry of the ocean. That's what's exciting. And again, we have not done this yet. We have just demonstrated that things like these are possible. But much of this data has not been incorporated because we don't have first principle arguments. And I think I'll defer a little bit. When we do the climate change discussion, then we'll cover up what are our moral responsibilities and all of those other things, because you could argue that might be the most frugal technology if we took to bicycles. And it goes back to Erica's question of upstream versus downstream. And we'll talk a little bit about it. I think that's a very personal answer. Okay, I just want to see if there is any other burning question. Ananda, go ahead. And then could you also just introduce yourself?
Oh, yeah. My name is Ananda. I introduced myself earlier. I'm from Toronto, Canada. I have a multidisciplinary background of working in kitchens and on farms and helping to do visual communications for open science hardware projects. But a burning question that I have is, I feel like it can vary from project to project, but a conversation that I've had with other sort of citizen science organizers is like, how do you go beyond just like using people and communities as points of data collection, and like what are the sort of points afterwards for putting in action to create policy or to sort of activate? Yeah.
No, I get the question. I'm going to defer it to another lecture that's coming up. We will have a dedicated lecture around how to build communities around ideas, and what is the role of those communities to begin with. There are certain sets of people. This will cover a little bit of what I often call the ethics of citizen science, where you could see that citizens are just a data collection point for certain sets of scientists or a certain set of a question, or the other way around, where empowering citizens to really be able to do science, to be the champions that need to be the voices that will cause change. And everybody takes a different set of a perspective based on the goals. And we'll cover a lot of this just from our own examples. I think, one of the challenges about all of this is that we can look back at the past and we can share what worked and what did not work from the sets of examples. But eventually people have to build their own sets of ethics internally as well. So I think my plan was to do this lecture in very end of the class. But if people are interested on Discord, what I will do is I will post the list of every single lecture that we are planning. And of course, there will be lots of unplanned time, and people can upvote and downvote if you want to hear about a certain content sooner, because I want you to get started on your projects now. Although you will be hearing about the content a little bit later. So I want to cover some of the problem spaces and some of the solutions a little bit early. And then many of these philosophical discussions as well, which are incredibly important, because clearly this class is not about technology. It is about causing change and using affordability and accessibility as a lens. So we'll cover that at some point of time. And I think to give you a preview, there is no right answer. It really does depend on what you're trying to do. And then we have a few handful examples of some fantastic things to think about. I mean, imagine, I was just reading a few sets of articles on the history of labor and union organizers. And you start thinking about what could we learn from them? How did they actually cause change to the largest corporations? And we could apply that to a citizen science framework. And this is just one of many examples. Okay, so in the last 10 minutes, I want to share one project that I'm quite excited about. We will bring that out in the lab session, which will be on Friday, which is associated with our response to this notion of frugal oceanography. First of all, I think one of the things that has happened for us as a lab is just spending a lot more time on research vessels. So this is Endeavor. There's a few students in the lab. And one of the things that I'm wondering if anybody remembers this boat. If some of you follow internet memes, this is Boaty McBoatface. I think if somebody's from the UK, they can explain the joke better. But the UK government was about to announce a research vessel for oceanography, and they did a voting program that anybody could write and say what they would like to vote. And of course, people being people, the boat name that the community at large chose was Boaty McBoatface. The government rejected that name, and now it's called the David Attenborough vessel. I am not so sure whether it was a good idea or not. But the reason I show this picture is because I spend certain amount of time in boats like these. This is Kilo Moana. This is from the NSF. And one of the perspectives is that it costs roughly around $50,000 to $70,000 per day, which is the operational cost. So if I have a cruise that is going to be measuring and mapping certain sets of things, which, for example, one of my students just went on the Palmer to Antarctica, Nathaniel B. Palmer. This is an icebreaker. It's even more expensive. You have to do a certain set of a survey, and you can literally be a month out there, and we're talking about millions of dollars of these. And the cost of measuring the ocean is incredible. And so in that perspective, a couple of years ago, we started thinking about how do we utilize and engage sailors and people that are passionate. So I'm just going to show you a 1-minute video of an individual that I met. He is a solo sailor from France. He travels around the world. But the realization was that there are 10 to 15,000 sailboats at sea at all times. And I'm not talking about fancy yachts and rich people buying their boats and keeping them on dock basically the entire time. I'm talking about people that deeply care about the ocean. And this is one example. He sent me this footage, and this inspired us to thinking about, could we instrument these sailors? He did sail with a chicken. That's his protein source. Not the chicken, just the eggs. The chicken is a very good friend of Gurek. Yeah, so the reason I like showing this video, he's currently, at that time he was in the, I think close to the Arctic. He probably spends around 11 months out at sea, and he'll dock from time to time. And there are literally thousands of individuals like Gurek, not as extreme as Gurek, but clearly a significant portion. And around the same time, we started thinking about this. I also met Columban, another individual that was starting to think about this notion of citizen science. And this is what led to the project, which we call Planktoscope. And a student who was himself a sailor joined the lab. So Columban had been working on this notion of getting sailors to do genomic sequencing, training literally what he calls citizen scientists. And they demonstrated that using this type of an approach, a very little hardware, you can actually map and get the kind of sequencing data that people are often used to seeing from traditional research vessels. And around that time, we started thinking about the context of imaging for sailors, and that really led to this project, which we call Planktoscope. Thibaut, who himself was a sailor, joined from France and spent several years with us, and Hong Chan and a couple other people. And we built the first version for Gurek's boat, and we wanted to see, could we actually instrument for a couple $100 an instrument that could measure the plankton in the ocean as Gurek goes around. You can see this is Gurek's trip. He left from France, he went down all the way. Actually, no, he went France, go up, he crossed. I think he's the youngest person to cross this Canadian trans-channel, go all the way down. He stopped in San Francisco. So this point of the journey is exactly that photograph. We got to spend some time on his boat. Then he went all the way down Antarctica. All kinds of crazy things happened here. Some of you know this is one of the dangerous parts of the ocean, and then he returned back to France. But one of the things we were able to do is really think about a type of an instrument that we could build completely in a modular way. And I'm not going to go into the details of this, but that's what became Planktoscope after many iterations. So if you go to this website now, planktoscope.org, you can see this instrument. It's built around an open source framework. It has all the components of Frugal optics that we talked about. It uses lenses that are called M12 lenses from essentially very simple imaging cameras in reverse configuration. It uses Raspberry Pi. It uses a PiCam module, has a very simple pump, and it has very simple focusing actuators. What it allows us to do is flow through the sample and then essentially continuously image. So I'll just show you the data that comes out of this. And then there is a lot of work that we have done at this point to do machine learning on this. So this, for example, is the kind of data we segment, and then after some amount of work, we can essentially put all of this is in a framework of identifying organisms. So this is, you can think of it as a digital version of the continuous plankton recorder. One of the exciting parts of this is we've shared this with the world at this point, all the instructions of how to build it. If any of you are interested, you can literally build it. Are online. We have demonstrated quantitatively that we can collect. So on the left is Planktoscope and on the right is FlowCam. FlowCam can cost anywhere between $50,000 to $100,000. And we can do a one-to-one match to an extent that one of these pictures is from FlowCam and the other one is from Planktoscope. And that's exciting because sometimes in this Frugal kind of a context framework, it is not about letting go of performance, but really building and designing around a different principle. Flow cams are triggered by a laser and they have a very different approach on how they image. We do very wide field of view imaging, but we eventually can arrive at the same results. That's really the bill of materials for Planktoscope is around $500 currently. And the first flow cam is roughly around $60,000. This is what that kit looks like. And I think the comment that I want to make is much of the instructions, when we posted the instructions, again, people around the world started building it. There's roughly, I would say, 30 to 40 replicas of these instruments that are out there, and people are starting to do a lot of different things with it. I'll bring a couple of Planktoscope in the next Frugal Optics session at the lab session on Friday. But I literally went to the website just today morning and I was looking, and I know if some of you are connecting from different places, we could look where is the closest Planktoscope right next to you. So if I go to planktoscope.org, I can go in and this is the current community, for example. So I'll zoom out just to give a context. And I think, you know, what's really fun is, of course, there are certain sets of adoptions that happen immediately. There's a lot of marine science community in Europe, so they have been the first adopters. And one of the things that we are starting to do now, and this is also, again, the purpose of the class, is to find broader communities around the world and make sure that they have a chance to access a tool like this. Okay, so that's all I wanted to say about a project. You will hear more about this when we connect at the lab session. But the purpose and the roots of this project are really driven by the type of a challenge that I described. There still remain incredibly number of challenge. To an extent, we've been thinking quite a lot about creating a group that's very specifically focused on the ocean itself. So I know I'm already over time. We can hang out for a few more minutes. For people who have to leave, don't worry about it. We'll catch up on Friday, but I'm happy to take a few more questions as well. And especially now would be a good time if some of you who are joining and were a little bit late when you joined, if you have any logistics questions too, from the global side of the story, this would also be a good time to ask. And, oh, go ahead, Deborah, yes.
Just checking in, so your lab on Friday, that's not available, like we can't zoom in and watch it?
We can zoom in, yes, there is a separate Zoom session for that. We will just have a Zoom running. And from time to time, we will try to have the camera pointing on a table. I think it's not going to be the same. No. I wish, Tyler, we should look if there is an avatar. You know what I'm talking about? That little robot that you can move around and people can control. We don't have one of those, but I think that's one of the downsides that's going to be, but we will be recording the sessions and we'll also be uploading that too. But there will just be a Zoom session running on the table. And we often, for the lab sessions, we start the first 5-10 minutes, we give a preview, which is associated with kind of like a talk.
Thank you. Thanks so much for trying to include us. I really appreciate it.
Yeah, I think we're very excited. And I think 97 people are in the global cohort, and we literally sent the email a few hours ago. So the fact that you all read the email and you're online means a lot to us as the teaching staff. And one of the things that I'm very excited about, which is in the past, we have seen an incredible intermixing of sets of ideas, and almost all the teams have people spread across, including the local Stanford cohort as well. So, I think you should participate. My hope is you really participate as a full-on individual. There is no watching from the sidelines when your house is burning Frugal Science. It really is. I mean, I'm hoping that you see this as an opportunity to engage. I mean, already I heard a soil scientist, I heard a food activist, all of you. I know Deborah is an incredible organizer in the context of thinking about how to take cell biology in these societal directions. And you bring as much to the table as we bring to the table. So I think I will lean on all of you. That's why I would love to get to know people better. Share your skills. Every lab session, we have a teach me anything session. You could literally teach people what food we should be eating, or how to convince other people to be thinking about food choices, or literally anything. There are no bounds around it, as long as it fits the ethical and respectful criteria of the class, because we are really trying to build and share skills as well.
And as a quick thing to hop in, I think maybe for the lab sessions, Manu, I'm thinking of trying during the actual hands-on portion. It could be a job that one of us, or even one of the students, depending on who's interested, could kind of do like a live stream version, like a vlog almost of hey, I'm going to put my camera into the scope. You guys can see
it's funny. I just learned today that when you share screen, there is a way to share an iPhone. So if you click on your share screen, you can see share iPhone, and then you're just in a video mode. And that might actually be fantastic, at least the first half an hour. So all the tools that are on the table, people get a chance. And then as people make friends, we will see that the team members, and they might have a team member that's remote, they will convince them to do this more and more and just really be chatting. Discord, I know, also has certain things like this, but Zoom has this. I think that would be fantastic, and especially because the timing is really awkward. I already know many of you are already late, and India is really the worst timing, and it's just right in the middle of the night. I don't know. Anandita, are you connecting from India by any chance?
Yes, I'm connecting from India.
Yeah. What time is there right now?
It's 5:21 in the morning.
I am so sorry. But
Right.
do you want to just introduce yourself? Just
Yes, so I'm a PhD student in mechanical engineering at IIT Kharagpur in India, and I work on electrokinetic flows and basically try to develop systems for point of care services. So
no, that's fantastic. Yeah, thank you for connecting. Oscar, did you get a chance to introduce yourself?
Hi, everybody. I'm in Mexico, Monterrey, Mexico. Here is about almost 7 p.m. 7 p.m. Yeah, I am a computer engineer oriented to software more than electric. And I am also an educator, a teacher. And I would like to take some challenge. And I hope I can contribute to all this.
Mm-hmm. Yeah, I think not only you can hope, I know you can.
I hope so.
Yes. And then Danane, do you want to introduce yourself? I don't know if you got a chance to introduce yourself.
Actually, I was kind of lost when I entered the call. So when you call like people for introduction of a global community, I was like, what's that? I thought it was like a separate program or something like that. But thank you. First of all, thank you very much for all the effort that you put on introducing all of us, like that we are not Stanford students, into this. It's a very good opportunity. I'm very excited about that. And well, really briefly, I'm Danai, and my last name is a two-word last name, so it's Chipocoaro. I'm from Peru, but I'm currently studying a PhD in material science and engineering at Georgia Tech. So yeah, thank you very much for the course. I'm really excited about sharing ideas and trying to learn more, because like I have a background in energy engineering. I have a background in, well, I'm studying now material science and engineering, but I'm not very familiar with biology and related stuff, but I'm really interested in sustainability in general. So I think that this is a great starting point that all of you can connect and share ideas in general.
No, that's fantastic. And then we will see all of you on Discord. Please do take a moment. It might feel overwhelming. There's a lot of conversations. Discord is our continuous 24/7 way of communicating, but you get used to it and you make the kinds of connections. So there is an introduce yourself channel, and then you can just write in. I have to head out to a PhD defense. So I think I'm going to run. But most of the time after the class, we will also have just an open-ended time for conversations itself. And I think, Tyler, you have the recording, so you should just post that, so at least the students that haven't had the chance to see their emails as yet get a chance to see this.
Yep, sounds perfect.
Bye, everyone. Thank you so much for connecting.
Thank you so much for joining, everybody.